As of the end of class last time, we had taken a molecule of glucose, broken it down into 2 pyruvate molecules, generated a little bit of ATP and some reducing equivalents. And where we finished was on the decision for cells to undergo aerobic versus aerobic respiration or anaerobic fermentation. Talked about the different redox reactions that take place along each one of those paths. We will pick up today on the aerobic respiration part, which, to remind, is generating some ATP, generating some NADH, and the pyruvate. And we're going to move now out of the cytosol. I don't think I emphasized this in lecture, but this is where glycolysis takes place. In fact, the evidence is that it takes place in certain microdomains in the cytosol where there are clusters of these metabolic enzymes, the glycolytic enzymes that we discussed last time. Aerobic respiration occurs within the mitochondria. And so everything that we'll touch on today is occurring in this organelle. And by way of introduction to the organelle, we should speak about some of the different sub-regions, subdomains in it, because they play a crucial role in the function of the mitochondria during aerobic respiration. Mitochondria is a double membrane organelle. Outer membrane, inner membrane, and then the space in between the 2 membranes is called the intermembrane space. On the very interior of the mitochondria is called the matrix, mitochondrial matrix. And all of the immediate steps in aerobic respiration that we'll talk about today are taking place in the matrix. The very last step of aerobic respiration, that is what generates the boatloads of ATP at the end of this metabolic process, involves the intermembrane space and a particular pump run in reverse that crosses the mitochondrial inner membrane and allows for the flow of protons, hydrogen ions, between the intermembrane space and the matrix. And the flow of protons back and forth across that the mitochondrial inner membrane into the intermembrane space is crucial to allow cells to undergo ATP synthesis upon aerobic respiration. I'll also point out here, because we'll get to it if time permits, you'll just barely be able to see triglycerides here on the upper portion. This is also the portion of metabolism where fats feed into metabolism. We'll come back to that after we cover aerobic respiration. Aerobic respiration takes place in eukaryotes because eukaryotes have mitochondria. This is the reason why prokaryotes only can undergo glycolysis or some of these fermentative reactions. And aerobic respiration overall can be broken down into 2 main components. The first has a number of names. We'll call it the tricarboxylic acid cycle, or TCA cycle. Some people also think that TCA stands for the citric acid cycle. Maybe it does. That's another name for this metabolic pathway. It also can be called the Krebs cycle, named after Sir Edwin Krebs, who helped to discover this metabolic pathway. The TCA cycle serves 2 roles broadly in metabolism. The first is to generate more reducing equivalents, such as NADH and another coenzyme that I'll describe in a little bit. And it also can be the point at which amino acid metabolites and metabolic byproducts feed into central metabolism, like we're describing here for glucose, pyruvate, and acetyl-CoA. The second portion of aerobic respiration is called oxidative phosphorylation. Another synonym for that is the electron transport chain. It's called that because this is the portion of aerobic respiration where we cache in all of the electrons stored up in reducing equivalents such as NADH. That involves oxidation of all of the reducing equivalents that we've described up to this point. So electrons are going to be transferred across a number of multi-protein complexes. And then the phosphorylation, what's getting phosphorylated is ADP to create ATP. And that's done by using a chemical gradient of those protons across the mitochondrial inner membrane. Just like in glycolysis, TCA has a lot of intermediates, a lot of enzymes that are a mouthful. We're going to emphasize the energetics of it, inputs and the outputs, mass balances, as well as the byproducts of it that are relevant for metabolism of other biomolecules or for energy requirements in the cell. Overall, the TCA cycle largely generates more of these reducing equivalents, reduced NAD in the form of NADH, and then another coenzyme that I will show shortly called FADH2. It's a cycle, unlike glycolysis, that runs top to bottom. And so I'll also try to emphasize where the carbons are moving as we go around the cycle. Several of them will get released in the form of carbon dioxide. But the main scaffold, the tricarboxylic acids, persist as you run around that cycle. As I already said, it's yielding CO2 as the byproduct, and it's also consuming water to achieve several of the metabolic steps along the cycle. Where we stopped last time was at this entry point into the TCA cycle. To remind, we had 2 molecules of pyruvate coming out of glycolysis, glucose cut in 2, so 2 3-carbon units. And it was a pyruvate dehydrogenase that performs another oxidation reaction that releases the CO2. So this is the first CO2 exhaled, if you will, from metabolism. This step is irreversible. You're not going to get that CO2 back and make pyruvate. And so as you might imagine, there are multiple allosteric controls on pyruvate dehydrogenase related to upstream metabolites of glycolysis, and then also downstream products of the TCA cycle. After this, we mentioned this briefly, we'll have more to say on this in a moment, we form this molecule called acetyl-CoA, 2 of the 3 carbons from pyruvate covalently bound to this coenzyme A. And it's that acetyl-CoA which feeds into the TCA cycle, as we'll describe. As we run around, I'll just highlight here on a high level, there are other CO2 evolving steps. Those are the release of another 2 carbons of the 3, so 1, 2, 3 from the pyruvate that came into the cycle. Those enzymes also are irreversible. You won't get that CO2 back from the atmosphere. And they're also subject to allosteric control by metabolites upstream in the cycle, as well as other small molecules in the cell. Say a little bit more about coenzyme A. Really the function that it serves inside the cell is to bind or transfer 2 carbon chains within the cell. First place you're going to see it is in that entry point into the TCA cycle, and those 2 carbon chains are acetyl groups, as shown up here. The structure of coenzyme A, some of these things should look familiar. Ribose sugar, adenine base, phosphate bridge. The only odd thing or new thing is this pantothenic acid, another one of those vitamins that if you take something in the morning, that's one of the things they'll give you, or and it's helpful with the synthesis of coenzyme A. We will see this first in TCA cycle, but I'll just mention it here so that you're ready to see it again. This also plays a crucial role in the catabolism and anabolism of fats by virtue of these 2-carbon acetyl groups that you can either stitch together in the form of a fatty acid, or you can break down through an oxidative process that we'll describe in a moment. I already talked about entry. Pyruvate, the dehydrogenase, release of a carbon dioxide, generation of a reducing equivalent to yield an acetyl-loaded CoA molecule, acetyl-CoA, to go into the TCA cycle. The next thing that happens once into the TCA cycle is that those 2 carbons will get transferred to a very central metabolite in the TCA cycle called oxaloacetate to yield a metabolic byproduct called citrate. This is the first place where one encounters 3 carboxylic acids. So 2 carboxylic acids here that came from oxaloacetate, and now the additional acetyl group transferred over as an acetic acid onto the oxaloacetate to yield citrate. So 1 carboxylic acid, 2, and 3. This takes water for it to accomplish. This step is also irreversible, subject to allosteric control. And we'll keep track of the 2 old CO2s here, which are the ones that will actually get released in the form of CO2 as we go around the TCA cycle, as well as the 2 new ones that came in from the acetyl-CoA as output from glycolysis. We're just going to go around the bend here. So we go around, there are multiple dehydrogenation steps here, catalyzed by dehydrogenases. Those are yielding the reducing equivalents, NADH, again, times 2, releasing CO2, again, times 2. So there's some parallels between the pyruvate dehydrogenase and these isocitrate dehydrogenases, alpha-ketoglutarate dehydrogenases, lots of similarities there. There's another intermediate step here involving another set of enzymes where Coenzyme A is involved again. And this is generating a nucleoside triphosphate, GTP, a form of substrate level phosphorylation, except involving GTP, generation of GTP instead of ATP. And when we do the accounting for the metabolic yield and TCA cycle and oxidative phosphorylation, we'll count this GTP as an ATP because there are enzymes inside the cell called dinucleotide kinases that will take in this case, a GTP and an ADP, and then catalyze the phosphate transfer to yield a GDP and ATP. And they likewise can interchange back and forth between the 2 states as a function of the products and reactants, the overall concentration of the 2 nucleoside triphosphates inside the cell. At this point in the TCA cycle, all of the carbons, as far as carbon accounting, have been released from the metabolic pathway. We lost 1 on the entry into the TCA cycle, lost the second one, lost the third one here. So that's where all the carbons go. But I'll note that the red carbons, the 2 that came in from the TCA cycle, are still in the pathway. They will continue to iterate around, and then they eventually will become old carbons, lost as they go around the loop a couple more times. When we finish the loop, turning around the bend, we have another dehydrogenation reaction here no CO2 released this time, and a different coenzyme involved. This coenzyme FAD, so stands for flavin adenine dinucleotide, chemical structure coming up in a moment. And its reduced form is FADH2. So no change in the charge, but it'll pick up 2 protons and 2 electrons to yield a reduced state. And then there's one final dehydrogenation reaction from malate to oxaloacetate, generates yet another reducing equivalent of NADH. And now we're back to oxaloacetate, the same TCA intermediate that we started with before the acetyl-CoA was conjugated to it. I'll also remind oxaloacetate came up very briefly at the end of the last class when we were talking about gluconeogenesis, the way to get from pyruvate back up to phosphoenolpyruvate, the terminal step that would ordinarily be catalyzed by pyruvate kinase and glycolysis, needs to go through an oxaloacetate intermediate to be able to get up to that phosphoenolpyruvate metabolite so that gluconeogenesis can occur. So oxaloacetate really central in a number of different metabolic pathways inside the cell. Here's FAD, back and forth between FADH2 on the right, another adenine base, another ribose, another phosphate bridge. All that stuff's identical to NAD. All right? The only thing that's different is rather than that nicotinamide group up on the top here in the schematic, we have riboflavin, another vitamin. Right? It's required. The reason why we need these is because we need to have certain concentrations of these coenzymes in the cell for things like TCA cycle to occur. And all the modifications are occurring here on these conjugated double bonds. So the TCA cycle completes the evolution of the carbons, the 6 carbons that we started with, into 6 CO2s. The entry point into it is with this acetyl-CoA. We already said multiple waters, multiple NADs, 1 FAD, and here for the accounting, we'll take that GTP that's generated by substrate level phosphorylation and count it as an ATP. So ADP to ATP, reducing equivalent of FADH, 3 NADHs with 2 CO2s released. This is focusing on just the cycle and focusing on just one pyruvate molecule. This all, when we go all the way up to the beginning from glucose, happens 2 times. So we have to multiply these all by 2. And if we combine this with the metabolic outputs of glycolysis and the entry pathway, here's where we currently stand. We had 2 net ATPs generated by substrate level phosphorylation in glycolysis. We made 4 but took away 2 during the substrate preparation. We generated 2 additional ones in the form of GTP converting to ATP. And then the NADHs, we had 2 from glycolysis, 2 from entry into the TCA cycle, and then 6, 3 times 2 in the TCA cycle, and to yield 10 reducing equivalents in NADH. Finally, for each one of the pyruvate molecules, or the acetyl-CoAs formally that go into the TCA cycle, we get 1 reducing equivalent of FADH. There are 2 of those per glucose molecule, and now we're at 2 FADH2s. All the numbers here, if you're looking from raw ATP generation, still are unremarkable because we have these mysterious reducing equivalents that have not been converted in any meaningful way as far as you're concerned into ATP. But it's at this next phase in aerobic respiration where all of these will become oxidized, release their electrons into other complexes on the mitochondrial membrane, and then provide a concentration gradient that will drive ATP synthesis. A lot of ATP synthesis through this process called oxidative phosphorylation. Questions on TCA cycle? Okay. Second phase, aerobic respiration, as I just said, is oxidative phosphorylation. It involves or also called the electron transport chain. The electron transport involves the fate of the electrons that have gotten picked up by the coenzymes NADH and FADH2. We will begin with NADH. There were a number of them, remember 10 per glucose molecule. And the acceptors and acceptors/donors of those electrons are a series of complexes that are in the mitochondrial inner membrane. I'm going to try to remember to do this, keep track of where the intermembrane space is and where the matrix is. I'm pretty sure they're all labeled, but sometimes it can be on the top and sometimes it can be on the bottom. And so please pay attention. In this schematic, the matrix, which is where all of the TCA cycle was taking place, is on the bottom here. Generates all the NADHs and FADH2s, they're right here. On the top is the intermembrane space. That's where, in a very high level, I introduced this idea that protons are building up there. The way that protons are building up there are through a series of multi-subunit protein complexes in the mitochondrial inner membrane called complex 1, 3, and 4 that will accept and donate electrons from those reducing equivalents and pass them through these other complexes on the mitochondrial inner membrane. At a very high level, I'll give you the gestalt of what's occurring here. As we said, NADH, again, just focusing on NADH, has the electrons that it picked up when it became reduced. It will now oxidize, lose its electrons, and transfer them to these complexes on the mitochondrial inner membrane. So we have an electron transfer from NADH into this complex 1. More to come on what occurs there, but just right now, I think complex 1 picks up the electrons. When complex 1 picks up those electrons, there are conformational changes that occur in that complex, which cause it to pump protons from the matrix into the intermembrane space. All the while, those electrons are percolating through the complex, and they eventually jump out on the other side of the complex within the membrane. There's another coenzyme involved that gets reduced. There are other iron sulfur clusters, cytochromes. These are all different proteins that can pick up electrons reversibly and then donate them to another biomolecule that's in the mitochondrial inner membrane. We're not going to dwell on the identity of every single one of them, but it's this handoff of electrons and the associated conformational changes in them when they become oxidized or when they become reduced that I want you to retain. As these complexes are gaining or losing electrons, there are conformational changes, and those conformational changes give rise to the movement of protons across the mitochondrial inner membrane. And depending upon the complex, there are certain numbers of protons that are pumped across that membrane. At the final stage of oxidative phosphorylation, this pump, this F0, F1 ATPase will open up, allow the protons to flow down that chemical gradient from the mitochondrial intermembrane space into the matrix, and it's through that downward flow of protons that ATP will be synthesized. So conformational changes, electrons occurring in the mitochondrial intermembrane, protons getting pumped, and you open up the spigot, protons go down, and then ATP is synthesized. I said this once before, but I'll say it again. At the very end of this electron transport, the final electron acceptor is oxygen. That's the O2 need. Right? Oxygen gets reduced into water, and that's the final fate of all of those electrons. from oxidative phosphorylation. We'll zoom in on complex 1 because it's one example where it's the biggest complex and also the one that we have the latest structural information on. So you can see this paper's not even yet 10 years old. This is what complex 1 looks like. Now to orient you, the top is the matrix and the bottom is the mitochondrial intermembrane space. So reducing equivalents, NADH, are up on the top of this schematic. And I said that NADH will lose its electrons and transfer them to complex 1. The electron acceptor on complex 1 is called flavin mononucleotide. Another coenzyme. All right, so it's going to get reversibly modified as a result of picking up those electrons. And flavin mononucleotide is bound to complex 1. As it becomes reduced, there will be conformational changes in all of complex 1. These are all individual subunits of complex 1 in the different colors. And within their structures, they have these so-called iron-sulfur clusters that reversibly undergo redox changes, states. So the electrons come off of flavin mononucleotide, jump over to one of the iron-sulfur clusters, that after it gets reduced, it gets octavied again, moves to another iron-sulfur cluster. And they percolate down this path through here, through complex 1. As the electrons are making their way through complex 1, all of the subunits in the complex are changing their conformations. And what occurs is that depending upon the exact position of the electrons in the complex, one can either have we should probably start here. In the reduced state, which is when complex 1 has picked up the electrons from NADH, there are pores open on the matrix side of complex 1. And those pores or pockets that are open on the side of the matrix, they bind protons. All right, so there's cavities on the 4 cavities on the side of the matrix bound to the protons. As the electrons move through and then eventually leave complex 1 entirely, that's the oxidation of complex 1. There's a conformational change of those pores so that they go from this position to this position. So it's binding, unbinding, binding, unbinding of the protons on one side and releasing it on the other side towards the mitochondrial intermembrane space. That gives the pumping action of complex 1. And the thinking is that the other complexes that we don't have as much structural detail on operate in the same way. Redox-dependent conformational changes, selective binding and unbinding of protons on one side of the mitochondrial intermembrane as opposed to the other, what gives rise to the pumping mechanism of these complexes. So same type of processes involved here with these protons. This cartoon is a little inaccurate and dated. This mechanism here should be inside complex 3. But this idea, there are different intermediates involved, but these conformational changes in the movement of protons is thematically similar. 4 protons pumped by complex 1 because of the 4 cavities that I showed in the crystal structure in the preceding slide. Similar movement, 4 protons in complex 3, and then 2 from complex 4. And I already said at the end, the final electrons, once they leave complex 4, go on to oxygen and become water. At this point, if people are hanging on, somebody might wonder what the heck happened to complex 2. We want 1, 3, 4. What kind of counting scheme is that? Complex 2 is involved, and it is involved with FADH2. The other reducing equivalent that I haven't mentioned up to this point. Complex 2 is the acceptor of the electrons from FADH2. In contrast to complex 1, complex 2 doesn't pump any protons. It simply accepts the electrons from FADH2 and then will transfer it to complexes 3 and then 3 to 4. You can see the distinctions here. So this respiratory complex here will accept the electrons. It can bind and recognize FADH2, but no pumping. Simply transfer into the later complexes. Thus, the net proton pumping outcome of an FADH2 is 4 fewer electrons compared to NADH because it does not engage complex 1. And we'll see why the proton accounting is important because it relates directly to the ATP generating capacity of the proton gradient. And where those protons come immediately to bear on ATP synthesis is with this pump run in reverse called the F1F0 ATP synthase. It has a motor generator characteristic to it. All right, so there's a stalk, there's a multi-subunit assembly here, there's a pore through which hydrogen ions can flow through, and it's called an ATP synthase or simply an ATPase because depending upon the concentration of the protons on the one face of the ATPase or on the other, this can either hydrolyze ATP and pump protons, or if the proton gradient is strong enough in the reverse direction, it can run backwards and allow the protons to flow down the concentration gradient, synthesizing ATP in the process. There are probably no homeowners in the audience, but if anyone knows what a heat pump is, like you know how you heat your homes around here? You have your air conditioner, you take your air conditioner in the summer and you run it in reverse and you take cold air in and you pump hot air out. Right? It's the same principle. It's just a motor running backwards using the same thermodynamics in the reversible process. The dynamics of the ATPase are as follows. You have conformational changes of the stalk and this rotor-like subunits of the ATPase, and it moves in these discrete steps, 1/3 of 360 degrees at a time, with each proton that, 3 protons going through is 1/3 of a rotation and yields 1 ATP synthesized from ADP. So 1 oxidative phosphorylation event needs 3 protons through. And now we have everything that we need to find the final net yield of oxidative phosphorylation and aerobic glucose metabolism from the top all the way down with oxidative phosphorylation. As we said, we get 10 protons moved up a gradient by virtue of complexes 1, 3, and 4 for NADH. For FADH2, it involves complexes 2, 3, and 4. We get 6 protons pumped. And we just said that as 3 protons flow down the gradient through the ATPase, that will synthesize 1 ATP. Thus, per molecule glucose, we said before, we have 10 reducing equivalents of NADH. So 10 times 10 is 100. 100 divided by 3 should be 33.3. Well, first of all, you can't make 0.3 of an ATP molecule. And then the reason why we have 30 here, this will come back in the next lecture, I believe, is that even if you said 33 ATPs synthesized assumes a perfect transfer of every electron from the NADH into complexes 1, 3, and 4, and every single one of those leading to ATP synthesis. That is so idealized as to be unrealistic. And in fact, biologically, the loss of electrons, the leak of electrons out of the electron transport chain is a fundamental facet of the electron chain and a key generating mechanism of so-called oxidative stress inside the cells. So as a result of aerobic respiration and aerobic metabolism, some electrons bleed off and they cause secondary damage inside the cell. Since evolution's time, we've needed to be able to tolerate, adapt to, and survive with. And so this 30 number from the NADH is to account the loss and the leakiness of electrons off of aerobic respiration. Finally, we have the FADH2s, so 2 times 6 is 12, divided by 3, we'll give that one 4. So all of the leakiness we put into the NADHs. I'll say more about that next time, but a lot of the leakiness occurs from the transfer of NADH specifically to complex 1 through that flavin mononucleotide, which is why we allocate the energetics this way. Now we have it all, everything that we need. From 1 molecule glucose, 2 net ATP from glycolysis, 2 ATP indirectly via GTP from the TCA cycle. All of the reducing equivalents that came from glycolysis, entry, and the TCA cycle are yielding 30 ATPs. And then the FADH2s, the reducing equivalents that were generated from the 2 pyruvates as they went around the TCA cycle, give another 4. Altogether, the stock number of ATP yield per molecule glucose is 38 ATPs. With the caveat that this is not a hard and fast number, but this is the number that metabolism people will work with when they are talking about glucose metabolism under aerobic conditions. Here's the complete reaction. Mass balance for carbons, hydrogens, and oxygens. Here's where the aerobic part comes into play. These are the CO2s that are relieved from the TCA cycle and the ATP produced. Questions about oxidative phosphorylation, electron transport chain? Looking a little woozy today. Right. There are a couple of, I'm going to say them odds and ends, more than odds and ends. They're kind of key considerations for this whole set of metabolic pathways to work. I talked about the TCA cycle occurring in the mitochondrial matrix. That's 2 membranes removed from the cytoplasm where glycolysis occurs. All of the intermediates here, whether you're talking about the coenzymes or the intermediates of the TCA cycle that are there, these are all large and polar species that don't just diffuse passively across the membranes. The cells, to be able to accommodate these different metabolic processes, have a whole set of dedicated transporters, membrane transporters, for different categories of metabolites. They fall into classes such as if you have 2 carboxylic acids or 3 carboxylic acids, if you're a nucleoside tri or diphosphate, if you're just free phosphate, pyruvate has its own dedicated carrier. Some of these are symporters, antiporters. They use different concentration gradients to allow things to occur. But the upshot is they don't just occur passively because they're all charged in large species that they can't go across the membrane. And this also occurs with some of the things that I'm going to talk about here with amino acids. Right? Some of amino acids also charged, large. They're going to need help moving across different membranes. Armed now with glycolysis, TCA cycle, and oxidative phosphorylation, we can now start looking how metabolites other than glucose or its byproducts feed into those core metabolic pathways. One that you heard about in the very first class of this course was amino acids used to make proteins through translation, all that stuff we did before. How do they fit in here? A couple of different ways, but one pathway that's featured very prominently is the TCA cycle. I'll give a couple of examples of this. If we take alanine and remove an amine, voila, we have pyruvate with the associated chemistry that goes along with that. If I take a TCA intermediate, such as alpha-ketoglutarate, and swap that with an amine, now I have glutamate. Same thing with oxaloacetate. Pick up an amine, swap out that carbonyl, and now we have aspartate. And in fact, these transamination reactions, I presented it in this way because the enzymes that catalyze this, in fact, take an alanine and an oxaloacetate, for example, and yield a pyruvate and an aspartate. So it's a sort of switcheroo of the amine and the carbonyls, or the same thing down on the bottom with glutamate and alpha-ketoglutarate. Perform this transamination reaction, and now you can swap out either metabolites in the TCA cycle to make amino acids if you need them, or if the TCA cycle is depleted, take amino acids and push them into the TCA cycle so the TCA cycle can operate. From the perspective of amino acids, if we're routing things into the TCA cycle, that would be amino acid catabolism. We talked about these terms before. Or conversely, if there's loads of the TCA cycle intermediates, they can be shuttled off in terms of specific amino acids for anabolic pathways, protein synthesis, cell growth, all those fun things. I should note that not every amino acid can just wedge its way into the TCA cycle. These are examples of so-called non-essential amino acids that we can synthesize de novo from other precursors. There are also essential amino acids that we cannot synthesize. We must get them entirely from our diet and from other organisms. Examples for us include things like phenylalanine, tryptophan, some of these hydrophobic amino acids, and including amino acids such as histidine. We can't make. We get them from the diet. But things such as alanines, serines, glutamates, those things we can just swap in by some of these biosynthetic reactions. Let's talk a little bit about fats. I'm going to get all these pieces because I can't talk about diets. You don't have all this. We just deal with fats. So fats are metabolized. I touched on this very briefly, like slide 1 today. This metabolism of fats occurs in the mitochondria. And it involves some things that you've heard. Acetyl-CoA is one example. But it's a special form of metabolism in the way in which long-chain fatty acids are broken down within the mitochondria. The pathway involves a series of oxidation reactions, dehydrogenation reactions of the long-chain fatty acid, 2 carbon units at a time. So here's an example of trying to make acetyl-CoAs by pruning off the long-chain fatty acid 2 carbons at a time. So what occurs? This is shown for completeness, so I can walk you through, but I'll just emphasize the critical portions here. We're going to start with a fat here, so long-chain fatty acid that's hooked up to a CoA molecule on the carboxylic acid end. So there's a fatty acid, carboxylic acid on the end. It will get conjugated to a coenzyme A molecule to form an acyl-CoA, oxidized by a dehydrogenase, generating a reducing equivalent here. We know what happens to that now, FADH2 going into electron transport. You have this is a water-dependent process. A couple of chemical steps you don't need to be aware of. There's another dehydrogenation reaction here involving NAD to NADH. And now you have 2 carbonyls next to one another. And the reason why this is called beta oxidation is that this beta carbon undergoes this oxidative, this gets oxidized, then it gets cleaved to generate an acetyl-CoA molecule, the rest of the long chain fatty acid with another CoA here on the carboxylic acid end, which gets you back up to the beginning, and you've now released an acetyl-CoA from the long chain fatty acid. So to summarize, if you have a long chain fatty acid, it's from the carboxylic acid end, gets broken down 2 carbons at a time through this process of beta oxidation, yielding reducing equivalents of NADH and FADH2, as well as 2-carbon acetyl-CoAs that can be metabolized by the TCA cycle, like we saw earlier today. All right, finally, we can take that piece, beta oxidation of fats. We can take the pieces that we covered last time and we covered today, glycolysis, TCA cycle, and begin to put these individual nutrients and metabolites together in a rough cartoon, which is shown on the left. And this will be the starting point of all the different diet strategies we'll have fun talking about after the break. But I want to round out the path involved with fats and long-chain fatty acids. At the very beginning, we're touching on obesity. I think we talked about circulating lipid levels, triglycerides. The free long-chain fatty acids aren't transported in the cell. They're carried around by these things called triglycerides. Has a glycerol backbone, and then the long-chain fatty acids, as shown on the right-hand side here. Lots of similarities to phospholipids, except there's no polar head group. It's just another long-chain fatty acid conjugated to the glycerol. These triglycerides can circulate in your blood. You go to the doctor, you get it checked. All right, this is something circulating lipid levels. When these are metabolized inside the cell, there's a class of triacylglyceride lipases, which cleave off one of these long-chain fatty acids. Then there are diacyl glycerol lipases that chop off a second one. And then there are monoacyl glycerol lipases that chop off the third one, yielding the long-chain fatty acids that then are the input for beta oxidation that I showed on the preceding slide, generating acetyl-CoA molecules that can be metabolized by the TCA cycle. At the end of that triglyceride breakdown, there's still the glycerol backbone that needs to be metabolized. It will get phosphorylated into glyceraldehyde 3-phosphate, which is another intermediate that you're aware of from glycolysis. I should say phosphorylated and oxidized. And now the glyceraldehyde 3-phosphate can go up to that preparation cleavage step of glycolysis and get processed down in that way. So triglycerides themselves have a fork in the road, glycerol backbone feeding into the upper portions of glycolysis, and the acetyl-CoA molecules feeding at the entry point into the TCA cycle. We'll speak more about this alternative pathway for acetyl-CoA metabolism, which arises when the TCA cycle is impaired, meaning you don't have enough carbons to shuttle around the TCA cycle. There's an alternative metabolic pathway to handle acetyl-CoA, and that will be brought to bear on some of the diet strategies we'll talk about tomorrow. Any final questions on the 3 or 4 metabolic pathways that I cruised through today? Enjoy your break. I will see you on Tuesday.